So I have one last function as now past president at this meeting. Listen, there's plenty of seats up front if anybody wants to come up and get a closer look at what's going to be happening. Although I think you can hear pretty well everywhere here. Putting on a meeting like this is an enormous amount of work, as you might imagine. And a lot of that work falls on the shoulders of the meeting chair and the program chair. And so, as is customary, we want to give our program chair, Han-Jung Jo, and our meeting chair, Han-Jung Jo, and our program chair, Julia Babensee, a token of our appreciation for all the work that they've done to put this together.
So it's my pleasure to welcome you to this plenary session that really captures the theme that we were looking to have in this meeting. And that is integrating academics, industry, and translation from discovery to implementation. And so this plenary symposium in translational biomedical engineering, models to practice, is really aimed at providing you with 3 examples of very effective and interesting translational models coming from different perspectives. And we have 3 speakers this afternoon. Each will be talking about their own experience in the translation of biomedical engineering from the perspectives of innovation development, education, and industry interactions with academia. And I would like to acknowledge the support of the Translational Research Institute for Biomedical Engineering and Science from Georgia Institute of Technology, and in particular the support of Barbara Boyens, that's her institute, her support in providing this plenary session for you this afternoon. So our first speaker is Donna Linger from the Wyss Institute. He is a professor of medicine and engineering at Harvard, and he is the director of the Wyss Institute. And he was just elected to the Institute of Medicine and is going to talk about a very unique model for the translation of technology. And so I'd like to ask him to come up and give his talk, which is Wyss Institute, a new model for innovation and technology translation.
Can people hear me in the back? Yes, good, okay. All right, so my story starts about 5 to 6 years ago. I was asked to co-chair a committee with Dave Mooney, a committee to envision the future of bioengineering across all of Harvard University and its affiliated hospitals. And to make a long story short, what we said is that bioengineering has been absolutely transformative for medicine over the past 20 years, 30 years, by taking engineering principles trying to solve a medical problem. But we were asked to look to the future, and what we saw is that the boundaries between disciplines are breaking down. Biologists are publishing in physics journals. Physicists have biology in the names of their journals. Computer scientists are interweaving in. We're beginning to uncover how nature builds from the nanoscale up. The boundary between living and non-living is breaking down. And so we think that the future is one where we're actually going to leverage biological design principles to develop new engineering innovations. And that's what we call biologically inspired engineering. Early on, we got the interest of a philanthropist, Hans-Jörg Wyss. He's Swiss, so it's pronounced Wyss. He came from industry. He's CEO of a major company. And he basically said, I know big companies, they're great at product development, but they cannot innovate. You cannot turn them. I can't turn my own company. I know academia, you guys are very inventive, but you make widgets, you publish papers, nothing ever happens. He said, I want to see a startup in the midst of the world's greatest academic environment. And so what I'm going to tell you about is what we think is a new model for innovation, collaboration, and technology translation. This is our logo. It's one of self-assembly because everything we do is through the process of self-organizing systems. We've self-organized this from the bottom up ourselves. The concept we came up with from the academic committee was to have an institute whose mission would be to transform healthcare industry and the environment by emulating the way nature builds. The reason that this is not just medical is that we felt that medicine and biology have scaled together for 300 years, but aerospace, manufacturing, energy, architecture haven't been touched. And the goals are simple. One, to continue to uncover nature's design principles, and the other is to engineer biologically inspired materials and devices. That's the heart of what we do, to revolutionize healthcare and enhance sustainability. So effectively, we started January 2009. It's 3 years and 10 months. We had the single largest gift in Harvard's history of $125 million. That's not for building. Harvard is contributing additional funds on a similar scale for building and administration. This is to be put to work. Focus on high-risk research, technology development, have to span academia and industry. We started by co-locating 14 faculty members from many institutions at a common site. We've grown to 325 full-time staff in 3.5 years. And we have over 750 acting collaborators that come back and forth. If anybody's done a startup, this is like 10 startups on steroids. We don't give faculty their own space unless they're new hires. We organize by collaboratory, where we basically have common spaces around projects, and we have people from different labs working together. And when that project ends, new things move in. One of the most novel things we've done is we recruited over 35 expert technical staff from industry with 10, 20 years in product development team management experience. We integrate with graduate students, undergrads, faculty, fellows as part of the team. This has totally changed the way we do things, and we have people from companies like Merck and Pfizer to Pratt & Whitney, Dane Kamen's medical device company, Sonos sound software, Microsoft Research, all collaborating together, a new type of interdisciplinary research. And we've structured ourselves to harness the entire Boston-Cambridge region, which we feel is critical in the time of international competitiveness. We're not in any school at Harvard. I report to the provost and to a board of directors. And it's an alliance between Harvard, its affiliated hospitals, which are separate competing institutions, as well as UMass Worcester Medical School, Boston University, Tufts, and we're continuing to expand to other Boston institutions. And our measures of success are novel for academia. We have the usual academic ones, but also the IP portfolio we assemble, corporate alliances, licensing agreements, startups, and having technologies in the product pipeline in 5 years, and having a near-term impact on our world. And I could just say that in 3.5 years, we're ahead on all of these points. We started by pulling together some of the most creative, entrepreneurial, and I like to say quirky faculty in our community, people like George Whitesides, George Church, Jim Collins from BU, Joanna Eisenberg, Dave Mooney, Dave Edwards, Mahadevan, for those of you, and younger faculty as well. And we've also been recruiting, so our newest recruit will start in January, which is Jennifer Lewis from, who has been at Urbana-Champaign in materials engineering. And then we brought in associate faculty from the bottom up because they were people that really made sense from a collaborative way to advance our efforts. So we basically took what we think of as the thought leaders in this emerging field, and we integrate them with the people from industry that we call our advanced technology team, who not only bring a commercial experience and product development experience, they're also our institutional memory. As an academic, it frustrates me when we get a big paper, we put in a patent, and then the postdoc gets a great job and the project ends. These guys are tasked with keeping that know-how internally. We bring in industrial and clinical collaborators extremely early on, and we have clinicians on our faculty as well, because particularly industry knows what the challenge really is in the marketplace, what competing technologies are coming down the pipeline, and where price targets are, and costs, and manufacturing. I recruited an executive management team from the startup world. They'd never been in academia because we wanted to create something that was, in effect, a new culture, and that's effectively what we've done. So the last slide, in terms of structure, we use an innovation funnel model where the broad part is the skunkworks of academia. That's what we do in academia well. Give people creative freedom, let them get grants, let them work between spaces in grants and do whatever they want. But then we have internal funding that's not, we don't give out grants, we fund platform activities, and I'll describe that in a minute, and we have staff who are paid for, and together they can begin to explore ideas. Once we come up with an idea and data that suggests it has commercial value, the people from industry help organize product development teams. And once we get feedback from industry that there's real interest, and we've got proof of principle and IP, then we develop what we call institute projects that we bring business development teams that are internal to try to advance this to the market through VC funding or in licensing or other industrial corporate investor sponsors. So the way we've organized it, as I began to say, is around platforms. We have 6, and these are effectively groups of people, faculty, staff, students, fellows, clinicians, and collaborators who are trying to jointly develop cutting-edge technological capabilities, not one thing, but a whole new approach that will enable a whole new wave of materials and devices. George Church said these should be cores that generate other cores in a technical core type of way, and that's already begun to happen. So in my time, I'm going to briefly give you a feel of the kind of range of what's going on, and we were tasked with taking on high-risk, high-potential payoff challenges that you know, NIH and current other government agencies, which are not as adventurous these days, would not take on. So the first we call anticipatory medical devices, and the idea is to develop portable wireless devices that sense life-threatening medical events before they happen and transmit signals that reboot or prevent disease. And we think this is the future of autonomous home healthcare. So the idea is you have be like a medical iPhone that would sense multiple dynamic physiological rhythms like gait, breathing, heart rate, and would be able to have algorithms that predict before you're going to fall or before you're going to stop breathing, and then would have some transdermal signal that would basically reboot or prevent. And amazingly, this we thought was the farthest out, but this we're farthest along on. So Jim Collins heads this with other faculty such as Ari Goldberger, David Petafar, et cetera. And they started by leveraging a technology he developed years ago that's based on stochastic resonance. This is a technology where if you give a random noisy, in this case vibratory signal, you could actually increase fidelity of a control of neuronal sensing. And he had data years ago that you can give 75-year-olds the balance of 25-year-olds by having them stand on a vibrating platform, like a mixer platform in the lab. What we have done is to translate that into shoe insoles that actually can be moved to the clinic, and I'll describe that in a minute. And the basic idea is if you have a nerve threshold, let's say here, and as you get older, your threshold moves up, you're less sensitive. If a signal was about here, and when you were young, you could sense it, and you're old, you can't. If you give a vibratory background, you basically signal, it's almost like a background energy that the input adds up to, and now it kicks things off. And this works across many different systems. And so David Petafar is actually at UMass Worcester Medical School. He took the same concept, he put it in a baby mattress, and he's published 15 human patients in the natal intensive care unit having this vibratory signal in the mattress prevents apnea of prematurity, significant suppression. He has new data looking at ventilation efficiency as well. Out of this program, because we just have staff that we've hired and now there's funds within the platform to play with, they've developed new sort of simple neuroassessment devices that are like iPad applications, just following a circle with a cursor with novel multi-scale algorithms that come out of the Goldberger lab that actually seem to be able to predict whether you have a concussion, for example, as well as flexible electronics. We have a gait control lab, many different orthotics. But to give you one example, when Jim brought this technology to our lab, it actually had been in a startup, but the startup died in the economic crunch of 2008. We took back the technology, worked with BU that owned the IP, developed they're one of our partner institutions, let us manage it. And this is an article from the Boston Globe business section, where it says the Wyss Institute taking risks on research industry won't do, because industry wouldn't refund that once it died. So we took it back. When we started, the company had gotten as far as what we call Bigfoot. They're about this thick, and they're about this big, and the battery is huge. And what we did is we basically believed in the technology, and in the intervening last 3 years, we've focused on things at this academic institute as such as reducing cost 100-fold, which we got from industry discussions saying what they needed to see. We've increased the runtime 2-fold, increased signal strength, reduced the size so that it could fit in conventional shoes, so to be an insole. And we've integrated the manufacturing from the insole industry with our technology. And we recently signed an agreement with a company, and we're starting human clinical trials. And if they work in the next year and a half, that will be a product, and we will get royalties. So that's one example of a platform. The next is one I actually had. It's called biomimetic microsystems. The idea is to engineer microchips containing living human cells that reconstitute organ-level functions, not cell or tissue, but organ-level functions for drug screening, diagnostic, toxicology, and therapeutic applications. And the idea was basically to accelerate drug development and replace animal testing. Now, the reason we did this is that the biggest challenge in the pharmaceutical industries, as well as cosmetics and chemical to a lesser degree, is that the development model is broken. In pharmaceutical industry, it takes over $2 million to test a single compound. It takes years to complete. Innumerable animal lives are lost. And then the results, more often than not, do not predict results in human clinical trials. And actually, the number of new drugs going into the clinic is actually going down, even though the dollars have gone up and up and up. So everyone knows that there's a need for new models. And we had a big breakthrough about 2 years ago that we call a human breathing lung on a chip. This is work of Dan Hunt and my group. And the idea was that the major functional unit of the lung is the air sac, the alveolar capillary interface. This is where you have gas exchange, infections, metastases, aerosol-based drug delivery, et cetera. And it's actually a simple structure. It's basically air, a single layer of epithelial cells, a flexible porous basement membrane, extracellular matrix, and then a single layer of capillary cells, and then blood. But what we also realized or took into account is that physiology is absolutely critical on mechanics, that the breathing motions of lung and the flow of the blood and the air-liquid interface are absolutely critical, and also the tissue-tissue interface that defines an organ, in this case endothelial, epithelial. So we set out to recapitulate this using microengineering techniques. And I'm going to show you a brief video that shows you how this works.
The lung on a chip is crystal clear, flexible, and about the size of a small computer memory stick. But it contains tiny hollow channels created using microchip fabrication techniques. A porous, flexible membrane separates the two channels at the center of the device. The opposite sides of the membrane are lined by human lung and capillary blood vessel cells. This mimics the arrangement of lung and blood vessel cells in the air sac of the lung. Application of cyclic suction in side channels makes the entire flexible sheet and cells stretch and relax rhythmically, just like our lung cells do when we breathe. In the lung on a chip device, air flows over the top of the human lung cells, and a liquid medium containing human white blood cells flows below the capillary cell layer. To test how well the lung on a chip device replicates the natural responses of living lungs, we introduced bacteria into the air channel to mimic an infection. And we introduced white blood cells to the blood channel. We then saw the white blood cells migrate across the capillary cell layer, through the pores of the central membrane, and into the airspace where they engulfed the bacteria. Here's a video that shows this response in real time viewed through the device. The tissue cells are not visible here, but we can see white blood cells flowing freely in the capillary channel of the device, just as they do in blood vessels of a healthy person. But when we infect the air channel by adding bacteria, the immune cells abruptly stick to the surfaces of the capillary cells on the opposite side of the membrane, located directly below the infection site. Here's a magnified view showing a migrating white blood cell making its way through the first capillary cell layer, wriggling through the pentagonal hole in the flexible membrane, and then moving out of focus to the other side. When viewed from the air channel, with all cells visible, you can see a round white blood cell popping up from below. Just like in a real lung infection, the white blood cells, which are now colored red, engulf and kill the green bacterial invaders.
So we've recently extended this, and we have a paper coming out in Science Translation Medicine in 2 weeks, where we actually developed a human disease model on a chip, in this case pulmonary edema induced by the cancer chemotherapeutic drug interleukin-2, which is known in humans that its dose-limiting toxicity is fluid transfer into the airspace and actually blood clot formation. I'm not going to show you the results, but we could see exactly this at the same dose and the same time course in this chip, and we actually can work, we identified drugs that actually come out of inhibiting mechanotransduction that actually prevent this response in our chip. And in the same issue, this is from a drug from GlaxoSmithKline, their researchers confirmed this works in animal models. And so we think this is sort of the proof of principle. You actually can begin to work with an entirely human organ-like structure to not only recapitulate function, but to do toxicity studies and drug development studies. We've also gone further. This actually translates, oh, I didn't say the most important part, which is that in these studies, breathing motions are absolutely critical for the responses we see. Without breathing, we only get about 15% of the edema response. Breathing increases it about fourfold, and when we do animal models, we see the exact same thing. The same thing with nanoparticle toxicities and absorption. Breathing is absolutely critical. This would not be seen with a transwell or a culture dish, or just cells on a chip. Recently, we developed a peristaltic gut on a chip, all human cells. We use Keiko-2 cells that people think of as very poorly differentiated, but when we put them in a device, we give them peristaltic-like motions and trickling type flow. We actually get villi forming, which you could see here and here. And we could put microbiome, living microbial cultures that come from the gut on their apical surface, and the cells are not only viable, but they get better barrier function, just like when you take probiotics. And so we're now looking at this as models for inflammatory bowel disease and drug development. I won't go into details, but we recently got a large DARPA grant where we're developing 10 different organs. Kit Parker's involved in this as well. And the idea is to link them microfluidically to create a human body on a chip, to develop an automated instrument to do PK/PD in these chips rather than in animal models. So that's second platform. Third is programmable nanomaterials. Idea is to create smart nanotechnologies for regenerative medicine and drug delivery. The idea is going from implantable devices to injectable programmable devices. Dave Mooney heads this. Dave has got a very exciting technology that is basically an artificial lymph node that is a cancer vaccine. He uses polymer technology to make a porous polymer, PLGA. He puts tumor extract in it, puts GM-CSF to recruit dendritic cells, and puts immune adjuvants under the skin in a tumor-bearing animal. It recruits dendritic cells, they get activated, they move back to the host lymph nodes, and they get a systemic response where they get 40 to 50% regression of melanoma in mice. They get much higher regression in glioblastoma. And this is so exciting compared to what's out there that we're just about to start a phase 1 human clinical trial at the Dana-Farber Cancer Institute, which is another partner institution. Now, we're developing all sorts of technologies. I want to just give you 2 other examples. One is from my group, and we focused on a major problem, which is vascular blockage. It's the major cause of death in man. Heart attack, stroke, pulmonary embolism, all due to blood clots in major vessels. Now, we thought there are good drugs out there, like clot-busting drugs, but only 4 to 5% of eligible people get them because of systemic bleeding side effects, bleeding into the brain. We thought if we could develop a way to target just to occlusion sites, that we might really have something exciting. Now, nature knows how to do this. Platelets target to occluded vessels or narrowed vessels due to atherosclerotic plaques due to increases in shear stress, a mechanical phenomenon that you can't have a genetic mutation to get away from its physical law. So we said if platelets do it, maybe we can mimic that. So we developed what we call platelet mimetics. We took 180 nanometer nanoparticles, PLGA. We made aggregates the size of a platelet, about 3 to 4 microns, and we coated them. Well, and the idea is that they're tuned so that they move around in the circulation as big aggregates under normal shear stress, below about 100 dynes per centimeter squared. But when they see an occlusion above 100 dynes, they break to single particles, which have a much lower drag force, and we coat them with molecules that basically make them bind more efficiently to clots. And the idea is that these are like a wet ball of sand. Hydrophobic forces are holding them together, and if you just shear a wet ball of sand, it falls to grains. It's the same thing with these particles. And so we coated them with TPA, tissue plasminogen activator. This is an FDA-approved drug. This bind integrates fibrin clots. It's already approved for these applications. And again, it's limited by bleeding side effects in humans. And in an in vitro model, we get beautiful degradation. But more importantly, we tested it in vivo in a mouse model, a pulmonary embolism model. The control, and this is labeled incorrectly in red here, all the animals die when you inject blood clots into the lung in 45 minutes. 100% die. If we just inject these, if we inject the blood clots and then we inject a bolus of these shear activated nanoparticles, 80% survive. But the most important thing is we use 1/100th the dose. If we gave that dose in a soluble form, there'd be no bleeding effect at all. So we're very excited about this technology, and we're actually trying to commercialize it in a startup right now. Another example of technology which we think is the future is DNA origami-based materials, the idea of building nanotherapeutics out of DNA, a biologically compatible material. And this is based on work that was first developed by Eric Winfrey of using a large circle of DNA, a DNA plasmids, having short strands, and then having them bind at multiple regions and folding up in a preordained shape like this rectangle. So the idea is, think of a circular plasmid of DNA. You know every nucleotide. You make a short DNA strand that half's complementary here, half there. When they bind, it makes a figure 8, like a staple. You add thousands of them. It can fold up into any shape you want. William Shee's taken this into the 3D. This is actually built out of DNA, this icosahedron, 100 nanometer scale. You literally have the plasmid. You have a computer design software we've developed, CAD/CAM software. You basically order the oligos, you shake them in a tube, and these things form. Peng Yin has adapted this so he could actually have small DNAs that self-assemble to make bigger things. He just had a science paper where he published, or a Nature paper where he published, he can build any shape letter you want at the nanometer scale. I think he has a paper in Science in Press where he can do this in 3D now using the same set of DNA oligos and just programming them like individual pixels to build absolutely anything you want. Now, I challenged the group early on to say, this is fantastic. Everybody in the field gets Science or Nature papers making toys, but could you do something useful? And Sean Douglas with George Church's group developed this cancer therapeutic. It's really a DNA robot, a programmable DNA robot at the nanoscale. He makes a hexagonal cage. The cage is held together by 2 little fingers that lock it like a purse clasps. And effectively, the locks are DNA aptamers that act like antibodies and can target to sites you want. And they only open when they bind to their target. And on the inside of it are ligands, in this case for death-inducing receptors on the cell surface, apoptosis-inducing receptors. When it's closed, the cells don't see it. When it's open, they kill. And by changing the locks, the combo locks with and/or not-if gates, you actually can program the robots to identify cells. And this is an example in their science paper last year. Each one of these targets a different tumor type. So these are truly programmable nanorobots, but they're made out of a biocompatible material. Next platform is synthetic biology. The idea is to use genome reengineering and directed evolution to create new materials, biomanufacturing plants, living cellular devices, and to reprogram living systems to provide new functions. This is the field of synthetic biology. George Church, Jim Collins, Pam Silver. They're making microbes for biofuel production. They're making automated directed evolution plants. They're decreasing the cost of DNA synthesis to enable those sorts of DNA nanotherapeutics. I'm not going to go details. And then Joanna Eisenberg heads adaptive material technologies to develop biomimetic materials and structures that dynamically adapt to their environment. And this is for architecture and energy. These are just simple nano-structured materials that are light transmission, color. And then she developed a surface called SLIPS that makes any solid surface into a non-stick surface by having a thin layer of oil on it. And these actually are self-healing. This is crude oil. She just made an injury in the surface, but because there's liquid, it refills injuries, and it still prevents adhesion. Now, one of the things about the Wyss, people thought we were nuts to go so broad, but I argued that by pursuing different disciplines, there'd be synergy by crisscrossing, and that has really turned out to be true. So, for example, we have a challenge to try to develop devices that prevent blood coagulation, and we took Joanna's slip surface, and this is conventional Teflon with fresh human blood, no anticoagulant, and you see it even on Teflon, it coagulates. But when we coat the surface with her SLIPS technology, as you'll see in a second, we take fresh human blood out of a donor, it just beads right off. And we've now done this on catheters, and we're getting liters per minute without any coagulation, without any chemicals added. Final platform is bio-inspired robotics. The idea is to develop biologically inspired robots that move and adapt like living organisms to create autonomous, self-assembling devices. The left is normal embryogenesis in the zebrafish. All these cells basically only deal with local cues, cell-cell contacts, forces, chemical in their local environment, and every time they robustly build the same embryo. If you cut them, they heal, and they still build the same embryo. On the right, you see a simulation with each little voxel is a robot that are acting in the same way, but they're programmed to build an airplane. And essentially, even though they say, oop, I forgot the nose cone, they self-heal and they complete it. This was done by Kasper Stoy and Radhika Nagpal when he was a visiting scholar. The point is that I really believe 100 years from now, we're going to manufacture like this. Many self-assembling units will work this way by simple rules. But we said, how do we start to get there? And that's what we said we'll try to do at the Wyss. And Radhika Nagpal has actually built now multiple robots that can work together. And this one is building a simple wall. You can imagine a sandbag wall. These can actually act collectively to build things that you want. At the same time, Rob Wood is really doing bio-inspired robotics, in this case flying insects, and they're beginning to fly. This is an old movie. It's about a year old now. They're much beyond this. But here you can see it's sort of failing, but it's sort of, on this one I think it actually makes it, and it's out of there, so it flies out. But the two of them are working together to actually use the programming for swarms of flying robots, which would be a great example of making this work. Out of these sorts of projects that are kind of out there come incredible technology fallout. So Rob, to make these, he used to hand-assemble insect robots. Now he developed pop-up manufacturing where you basically have single degree of freedom. This literally is laser cutting multiple layers of conducting, resisting, mechanical actuated materials, and you just pick it up at the end and you have a flying robot. They used to do it with tweezers under a microscope. And this could be used for any sort of medical device or toys or electromechanical type of machine. And I should say, Radhika had our first licensing deal with 1,000 self-organizing robots that use RFID and a flash to program them for educational purposes called Kilobot. So this is my last slide. Basically, I had to put this together in the summer. In addition to what I told you, at 3.5 years, starting from zero, in addition to all of the people and organization and recruitment, we've secured now over $110 million in grant funding. We've had over 425 publications with only 14 core faculty and 3 new recruits, most of whom just started a year ago. We've averaged 1 Science or Nature paper every month for 3.5 years, and we just had 4 last month. So you can't say we're not doing basic stuff as well. We're up to over 200 patent filings and multiple issued patents. We have 25 clinical studies and 2 human clinical trials about to start. We're at least 20 product development focused collaborations with industry. Again, at least 2 licensing agreements with 1 product being sold. And we have at least 4 startups in discussion. And so to end, I invite you to the website. We actually won the Webby Award this year, which is the Academy Award of the Internet for Science. Our competition was Wired Magazine and the NASA Space Propulsion Lab, among others. I think you'll enjoy it. But I hope you can see that this is something new. It's something that can work. And it's something that we need to do in many places, explore new models to really bridge this space between academia and industry, especially as investors get much more cautious and want more de-risking, not only scientific, but even commercial de-risking before they'll help a technology get out of the lab. So thank you so much. Do we have time for questions? Or? Yes, of course.
So we have a couple questions, if you can use the microphone. You can talk really loud.
You start looking, my friend. I didn't have it in the beginning either. Well, to be really candid, the money really allowed us to do things and have freedom from usual institutional internal control issues and budgets and so forth. And that is really the most critical part. If you have an institution that is forward-looking, then institutions have money for different things. It's just convincing them not to build a building and to put it into people. You know what I mean? That's an example. But there are a lot of people with money still out there who don't want to give money when institutions say, give us money because we're great. But they're very interested in giving money if you say, we're going to change the world and do something new and show we have a better way. So I think you have to ask. Thank you.
Oh, there's another one.
Yep.
Yep.
We actually think that we will have groups of people and projects moving out. Literally, people that were in industry, who are in a lab working on a project, is extremely attractive for an investor if they're going to do a startup. So we think there's going to be an outflow of people and new people coming in. I think we have to think about not doing the next iteration of what we've done, but think about major challenges so that it's still future-focused and high-risk. We're having a retreat. We have 550 people attending in a few weeks. And that's the theme for that is, where are the next big challenges? Just doing another cancer vaccine or another manipulation of this is not really what we want to have as a major focus. So we have to stay fresh. That's the responsibility of leadership and the faculty. Thank you.
So our next presenter will be Professor Paul Yock. He is a professor of bioengineering and medicine at Stanford University and director of the Biodesign program. And the title of his presentation will be Biodesign, Teaching Biomedical Engineering Innovation as a Discipline. And a little bit of background on him. He is a member of the National Academy of Engineering, and we're very interested in hearing about institutionalizing and teaching the biodesign. Thank you.
Thank you. Well, it's good to be here, and I want to pick up on the sense of innovation that Don was using, and financial disclosures. I won't be talking anything about products here where I have a financial conflict of interest. So the context for innovation that I want to talk about is a context that Harold Evans uses. I like a lot his description of innovation not being just innovation, just invention, it's invention put to use. So I've encountered, you have two folks who seem to be born innovators. These were a couple of my mentors, Tom Fogarty and John Simpson, who together account for hundreds of patents, millions of lives treated. People like this have a gift, but the point that I'm going to try to convince you of over the next few minutes is that although people like this have a gift, there's a process, a discipline in innovation that can be learned and practiced and perfected. Not a new idea. I think the most eloquent expression of this is from this book, How People Learn. It was a result of a committee from the National Research Council led by John Bransford, and he generated an interesting graph that I like a lot. He said, look, what we usually do in education is we take novices and give them expertise. Think about Ph.D.s in bioengineering. We make routine experts. And on the innovation axis, we tend to wait as a society until these brilliant people hiccup up in our midst. The issue being that sometimes they're a little bit rudderless. And Bransford said what we should be concentrating on is taking people up both axes, having experts who understand a process of innovation, and that's what I'd like to talk about. I think our field of biomedical engineering, by the way, has done the best job, at least recently, of the engineering disciplines in trying to work on training processes for adaptive expertise. This is from a meeting that we have every other year. There'll be one at the next BMES meeting of an ad hoc group called BMEIDEA. The acronym stands for Innovation, Design, and Entrepreneurship. This is over 100 universities that are experimenting with teaching techniques in this area. And there are some phenomenal examples of programs at the capstone level, at the master's level, and above. I'm going to focus in on the program at Stanford. We're one of the older programs. We're in our 12th year now. Tell you a little bit about how that has worked out and some of the outcomes. It's called the Program in Biodesign. It's part of a larger multidisciplinary initiative that goes across the university called BioX. And the premise is based on this. As you all know very well, in industry and in academia, we do a lot of technology push innovation, and there's absolutely nothing wrong with that. we've just seen examples of phenomenal technology platforms that will find really important applications. But we tend to underweight need pull innovation. And what I'd like to describe is the fact that there is a process of doing needs-focused innovation that can be taught as a kind of a discipline. We structure this in sort of 3 phases, but the important point is that we start out purely with an identification of needs. There's no technology at the start of this process. Let me tell you how this works using the example of the postdoctoral program we have that we call the Biodesign Fellowship. We have a national search for young people who are would-be innovators in the medical technology area, and we put together teams. This was a team from a few years ago that had a chemical engineer from Delhi and a surgeon from Yale, a mechanical engineer who came out of one of the companies to do this, and a freshly minted electrical engineer. We do 3 teams a year now. And each year, we put these teams into a clinical area of medicine. We change that every year. So this year, for example, we're working in neurology and neurosurgery. And what we do is we have the team, first thing they do after getting a little basic understanding about the area, is they go live in the hospital. And they live in the hospital for 2 months, and they don't come out of the hospital until they've found at least 200 clinical needs. These are opportunities which, if they were pursued, would have potentially an interesting technical solution, but they're just needs at this point. Then we go through a process, and this is the hard part of the process, of screening down those needs to the top few. And we use as our filtering criteria these 4 things. We look at the clinical impact, so how many patients are impacted by this in the country or worldwide, and how severe is the impact of that condition. Who are the stakeholders? So are the people who are going to be affected by a new technology aligned, the payers, the providers, the patients, or is there one of those groups who would inhibit the development of a technology in that area? What are the existing treatment options, and how well do they work, and how much do they cost? And importantly, what are the market characteristics? So we do a frank business analysis right at this point and say, is there enough of a market that an invention in this area could be supported either through a startup or through licensing to a company? So we winnow down that list of a couple of hundred to just a handful of needs that we understand really well. And we have a mantra in the program. We say a well-characterized need is the DNA of a good invention. So going forward with those needs, this is just an example. You can't read this. This is one of the screening algorithms that one of the teams developed. We have each team invent its own kind of screening weighting software function and has the parameters that I mentioned. We have a couple of other formalisms. One is we ask for each need that there be what we call a need statement. This is a 1-line mission statement about that need. So this one says we're looking for a safe, effective way to reduce the apnea and hypopnea episodes experienced by a patient with obstructive sleep apnea, hypopnea syndrome. That becomes the guiding light of the entire project. We spend a lot of time just understanding what the crisp mission for that need is. And then we develop a need specification, a set of criteria that a good invention has to have to meet that need. It's kind of like a product spec, except it's still at the need stage. So this takes 4.5, 5 months to get through this process to have a well-characterized needs. And finally, at that point, it's time to invent. And I like what this team said, and it's completely true, that given enough time, sugar, and caffeine, you will invent something. It is a luxury of the medical technology field that if you get the need right, you're pretty much guaranteed of being able to find a technology or a combination of technologies that will serve it. And I'd make the case that this is a different kind of innovation pathway than in pharmacology and drug discovery, for example. Okay, so from here on, we've got the needs. We use design methodology. We ask the teams to invent several different concepts for each need. They take those through prototyping, early stage testing. And then we do another kind of a trick, which is we make all of those concepts compete with each other through a second set of filters. And the second set of filters starts with intellectual property. So we haven't looked at intellectual property seriously until this point. And that's a philosophy we have in the program to not bring in intellectual property until here. We also look at the regulatory and reimbursement situations, and then the business model. Is it capital equipment? Is it disposable? Is it a startup? Is it a license to an existing business? And we use those filters to come up with one final concept out of all of these that's worth taking forward, that survives, that competes well with these others. So if we put this together, you see what we've done. We've started with a couple of hundred needs and had them compete with each other to come up with just the best needs, blow it out again into a number of concepts, have all of those concepts compete with each other to come up with just one final concept. So this is a methodology that we think treats the major disease that inventors have, and that is falling in love. You can easily fall in love with a need. There are a jillion needs out there. What you have to do is find the right need that, if you solve it, is worth solving and will get to patients in the marketplace. Same thing with your concepts. All of these concepts are cool ideas, but you really want the one that you're going to invest the next 10 years of your life in to go forward with. So this is a discipline that forces you to pick something that really is worth working on and having people spend their money on to develop. So that takes us to the final concept selection. And then there's a whole implementation phase. I'm not going to go through this in detail, and these are too tiny for you to read, but we talk about more advanced intellectual property strategy, clinical strategy, reimbursement, marketing and sales, all of these things that go into getting to the stage of licensing or having a business plan. There's a textbook that summarizes this, and a website actually that has some 15,000 links into useful areas, and that website is called Biodesign. So we also have a graduate student class besides this fellowship, undergraduate class, and have the luxury of having a lot of advisors from industry. The fellows meet somewhere around 150 different advisors during the course of the year. So putting all of this together, if you have smart young people working on real needs, of course, real technologies happen, and we've had a number of things go forward out of the program. In fact, we had our 26th company funded last month. Not all of these companies are going to survive, of course, but they're all alive at this point, and some of them actually have FDA-approved technologies. So at the end of each school year, we tally up, and this last year, there were a number of patients, 120+ thousand patients treated by technologies coming from these first-timers, from these students, 510 new jobs created in their startup companies. But our point is not to create products or create companies. The point is that there is a training process that you can learn as a young person. And the luxury about med tech is that the product development cycles are short enough. They're 8-10 year development cycles that these folks can do this multiple times during their career. So our whole thought is let's take people through the process once early on in a highly mentored environment, and then hopefully they will see where the potholes are and not make critical mistakes in the second and third and fourth times that they do this. Now, that's a rosy picture that I've given you. The reality is we're at a historic turning point, in terms of medical technology innovation in this country. And there's a lot of talk about a perfect storm, which actually is true. So we have a combination of three things going on. The FDA, for very understandable reasons, they're under enormous pressure from Congress and press, has gotten more conservative, more unpredictable in approving technologies. We have a reimbursement issue and a diminishing in venture funding in the area. This is just a trajectory of the PMA type FDA approvals for medical devices over the past several years. With respect to healthcare reform, the healthcare economists uniformly agree that the uptake of new technology is the biggest single driver of healthcare cost escalation in our country. So there is enormous pressure to slow down the uptake of new technologies. And that's what's going to come into play over the next few years in lots of ways. But one of the ways will be that Medicare is going to increasingly ask for real cost-effectiveness evidence, large-scale trials that shows that what the cost implications are. All a good thing to do. The trouble is that it prolongs a technology being taken up by years, and those years cost hundreds of millions of dollars. So that's getting harder too. No surprise, as a result, venture funding in the sector is going down. Again, you can't read this. I just want to give you the gestalt of these curves for funding and new deals created that have gone down in the last few years. In fact, this last quarter that we finished is one of the lowest quarters for medical technology investment and biotechnology that we've had in several years. All right, so it's a historic turning point. What do we do about that? At turning points like this, there is also great opportunity being created. And I think there are a couple of directions that are exciting for the students in the audience to think about. One of them is the opportunity to look for technologies that actually take costs out of the system. So if you look at the course of how we spend money on a given patient, the event that drives costs through the roof is hospitalization. If you can keep patients out of the hospital, and Don was talking about some technologies in this zone, that's the single most effective place where you can apply leverage to keep costs down. The second area is to try to shift the delivery of care from expensive providers to less expensive providers. So take a technology that a specialist or a intervention that a specialist is doing, allow a generalist to do it. Take something a general doctor is doing, allow a nurse to do it. Or best yet, take something that a nurse is doing and allow it to be done in the home by the patient or the family. Those are the key cost-saving mechanisms. Let's look at an example. So we had a team a few years ago who looked at this problem. If you have palpitations, skipped heartbeats, and you go to see an internist, the internist will listen to you. Nothing will be going on, of course, at the time you're in the doctor's office, and he or she will say, well, you know, it could be serious. I've got to send you to a cardiologist. Same thing in the cardiologist's office. Cardiologist sends you to a rhythm specialist. The rhythm specialist sends you to have one of these Holter monitors put on. You wear that for 2 weeks. The rhythm specialist gets the information back, communicates it back to the cardiologist, who talks back to the internist, who talks to you 4 weeks later and says, most of the time, there's no problem here, you're fine. But occasionally there is a problem, and you sure wish you had known that a couple of weeks earlier. And so this team designed a smart bandage with the idea that when you go into the internist's office, the internist should be able to reach in the drawer and put on a bandage that will record your heart rate. And you wear it for a week or 2 weeks, break off the memory unit, send it in like Netflix, and your internist within 24 hours gets an email back saying, everything's fine, or, you know what, need to see a cardiologist ASAP. So this is the team filing their provisional patent. Uday Kumar took it forward to run the company that is now called iRhythm Technologies, and the Zeopatch is the commercialization of this idea. Takes tremendous cost out of the system. You've eliminated 2 specialists, you've sped it up, you've used current technologies to increase the rate of information flow back to the patient. This is the kind of cost-saving technology that's going to be important over the next years. So the second and final direction is, of course, taking advantage of the fact that it's a global community, and with especially the BRIC, Brazil, Russia, India, China countries that are these emerging economies, that there's an engine here for value-driven innovation. If we zoom in on India for a second, the rate of growth of medical technology demand is somewhere between 10 and 15% per year in India. It's 1% in the United States. It's flat in Europe. And interestingly, in India, there's a major demographic shift going on. I love this picture from my colleague Raj Doshi showing some of the slums in the foreground and these high-rises emerging. This is exactly what's going on with the demographic. There's a massive shift of people, 140 million Indian nationals, who will move out of poverty into the middle class. At the same time, there's a expansion of chronic disease in India. Our own form of bioterrorism is going on here with developing coronary disease. Half of the world's heart attacks are going to happen in India in 5 years' time. So what does this mean? How does this come together? This powerful emerging middle class in India and China and these other developing nations are going to demand good technologies. Here's an example, a standard Medtronic pacemaker coming in, the dual chamber at about 10,000. There's a company in India now called Medived that has a $2,000 pacemaker, very functional pacemaker, and there's been a great feedback loop. So Medtronic has responded with this with a solid, substantial pacemaker and a new business opportunity that reduces the price for Indian nationals. GE has done a terrific job in India with a program they call In India for India, has developed this EKG scanner that is a very robust, heat-resistant, sturdy unit that does interpretations and has not only impacted the India market, but the other developing nations as well. So where does this go educationally for us as we think about what the mission is in training and innovation? Our take has been to try to find and help train in-country innovators as a start. So we have a program in India called Stanford India Biodesign. In fact, Amit has been here at this meeting. He and his colleagues started the first startup out of this program. It's based in Delhi. I'll give you one example of, again, how needs-finding, needs-based innovation can lead to interesting consequences. So this year's team was looking at trauma and orthopedic injuries. And there's a lot of motor vehicle trauma in India. And what the team observed in Delhi and a number of the other states in India, the governments have done a good job of deploying state-of-the-art ambulances and ambulance equipment. And so what they saw a number of times was the following scenario: motor vehicle accident, nice ambulance goes to the scene, picks up the patient. They put on a great splint, take the patient into the hospital parking lot. Gurney comes out of the hospital, out of the emergency room. Patient gets transferred to the gurney, and the ambulance crew takes the splint away because it's a $200 splint. It's the only one they own, and they can't afford to leave the splint with the patient. Not exactly good medical practice. And so the team very reasonably said, what we need here is a disposable splint. And they went ahead and developed a prototype, did manufacturing in a box plant in Delhi for 8 cents per splint, took this through a clinical trial at the All India Medical Institute. The entire cost of this development, so needs finding, prototyping, testing, clinical trial, was $5,000. a scale that we can't come close to here. So last slide here. This is, I think, a really exciting time for you all, the students emerging as innovators in the biomedical technology space, because I think there's this virtual cycle going on. We have increasing cost pressures, increasing barriers to new technology development in the US. On the other hand, in Asia and the other developing countries, we have these engines of expanding markets and an intrinsic natural focus on affordable technologies. And these come together in a really nice way, I think, to provide us all as we're training with the incentive and the experience to develop affordable technologies that we can bring back to the United States and bring an attitude to the US of inventing affordable technologies for our own patients. So I'm going to stop there. Thank you very much. Should we go ahead, or what do you think?
The next speaker is Jim Burns. He is the head of Sanofi Boston R&D Hub, and that has been recently acquired Genzyme, and he spent about 24 years of his career at Genzyme since he was a postdoc. Has been highly interactive with academia, and so we'll talk about those experiences and integrating a translational approach into product discovery and development.
Thank you, Julie. And I'm an engineer, but usually right about this time is when my engineering skills disappear, trying to get the presentation up. Let me try this. The arrow is missing. Go up to okay. There we go. All right. So my intention is to give you a little bit of the industry perspective in what we call translational medicine, translational sciences, and the industry-academic interactions. And not to put too fine of a point on it, but there's really one issue I wanted to talk about, and it's been discussed a little bit already. It has to do with ideation and generation of ideas, and I think it's something that's very often overlooked in the translational medicine area. So there is a problem. You might ask yourself, well, what is translational medicine, the medical technology area? What does it bring to medical technology? And what's the problem that we're trying to fix with translational medicine? Very often that's how in industry we think about things. What's the problem, and what do we do to try to fix it? And you've already heard that there's a productivity problem in industry, both in the pharma industry as well as in the med tech industry. I work for a company, I'm a bioengineer. I work for a biopharma company, work on medical devices as well as on drugs. So I see both sides to this. And there's a drop in productivity as well as innovation in the med tech area in terms of number of PMAs as well as 510Ks. It's costing more, and it's taking longer to get 510Ks and PMAs approved. And it's the same in the pharmaceutical side. So there are fewer drugs being approved, but it's costing more. And actually, to one of the questions that was asked earlier, think about that for a second. It's costing more, but number of approvals is coming down. So it's obviously throwing more money at a problem isn't the solution. It's ultimately how we innovate that's important. So There are other problems associated with product development and discovery and innovation. And some of this has already been covered. Not only is productivity going down, but the environment, the healthcare environment, has changed substantially in a very short period of time. I mean, one of the big things is that we have really an untenable situation in terms of healthcare costs. So today, about 17% of our gross national product is spent on healthcare. And it's growing, that amount we're spending on healthcare is growing faster than economic growth. I mean, it's just unsustainable. So we have to do things differently. We have to be smarter about how we develop new products. And so that's where translational medicine and translational sciences comes in. It's intended to translate new treatments into products better, more effectively, more efficiently. And as I was trying to think about my comments, I realized that, you know, as an engineer, I think of translational medicine, translational sciences as a process. And it's the process of taking ideas and transforming them into innovative products. You know, an invention or an idea is not innovation in and of itself, as Paul was just saying. It's only after it's been transformed into something that's actually different and useful that you really have innovation. A lot of translational sciences and medicine, though, focuses on the innovation process. You know, the trying to create clinical trials allow you to get an answer to a question faster. Trying to establish proof of concept or whether a product might be safe or not as quickly as you can. Trying to develop preclinical models that are more predictive of what you might see in the clinic. And all these make absolute sense. But there's another part that I think is neglected, which is the ideation part of translational medicine. And I'm going to ask you to try to prove a point in my next couple of slides, but on this slide, take a look at this slide and try to think what's common and different amongst the three examples here of innovation. You have John Lister, shown in this picture, was the father of modern surgery, angioplasty, and the discovery of statins. And Joseph Lister first used carbolic acid to try to prevent gangrene in patients because he observed, as most surgeons did at that time, that you did surgery and more often than not the wound got infected. What can we do about that? And he knew about Louis Pasteur's work, and that in particular with gangrene, there were things that you could do to try to prevent that, and one of it was perhaps use a chemical on the wound. And so he took that observation and started to think about what could I do about that. And that's where he came up with the idea of using carbolic acid to treat wounds and sterilize equipment. Because at that time in England, carbolic acid was being used to treat railroad ties to prevent them from rotting. Rotting skin with gangrene, rotting railroad ties, why not use the same treatment? And it worked. Angioplasty. Following angioplasty procedures with a balloon, you have a certain percentage of those vessels that reclose, and thus the observation, what do we do about that? You come up with stents. HMG co-reductase is an enzyme in the liver that's involved with synthesis of cholesterol. It was thought, well, maybe we can try to inhibit that enzyme to try to lower cholesterol. But what's different about these observations? Let me contrast innovation and translation of ideas between pharma and the med tech area. So ultimately, we want to get something that's useful to the patient, that's a product that comes through clinical trials. And that's where start to get into some differences between pharma and medical technology. So in pharma, like the statin experience, a scientist might read an article and get an idea and then start doing experiments and actually conduct a discovery program. In the medical technology area, there's a direct observation. You spend time with a surgeon or a physician, you get an idea of what the problem is, and you start to think about what can I do with that to solve that problem. You do that with business partners, engineers, scientists, and it's an iterative process. And this is something that's it's different than pharma, and it's something that is overlooked, I think, in translational sciences and translational medicines. There's another issue here now or challenge that we have, and I see this, which is that more often than not now, medical devices are, or very often anyway, are coupled also to drugs. So we have to somehow bring those paradigms together as well in terms of moving into clinical trials. So let me give you an example of this. This is from my own personal experience. Observation, something I was personally involved with many years ago. Observation that scar tissue forms after surgery. These are called post-surgical adhesions. What can we do about that? I spent countless hours in surgeries, cardiac surgeries, orthopedic surgeries, gynecologic surgeries, as well as abdominal surgery, trying to understand this problem, working with surgeons to try to figure out what would be a reasonable approach. And to make a long story short, we designed some ways of doing this, a bioabsorbable membrane, tested it in the clinic, and it ended up working. Today, that would be much harder to do for somebody in industry, to spend that amount of time with surgeons and then to go through this iterative process to try to turn that into something, an idea that you then design around and then transform into something that would be a real product. And it's harder for a lot of different reasons, and I won't go through all of them. And some of this has occurred because of the Affordable Care Act. There's a need for more transparency. Transparency is good, but the activation barrier for those interactions has increased. So what do we do about that? And that's where I think companies and academics and other groups have to start to partner more in different ways. We need to be able to have more open relationships. We already heard from Don on the Wyss Institute. I think academically, there needs to be closer alignment between engineers, scientists, and hospitals to try to help with this process of observing and translating that into something that's actually testable. Quite frankly, it's easier for you to do it than it is for us to do it in industry now. We need those ideas, and we need those ideas to mature a little bit further before it's something that we can act on. Additionally, companies need to change their innovation paradigm. So I'll touch a little bit on these. So this is something that wouldn't have happened a number of years ago. So I work for a company that works in the diabetes area, and we are going outside, as well as internally, to try to generate new ideas for a delivery device. So not only we're looking at things within our company, we're going to external design houses, academic groups, as well as crowdsourcing for ideas. Other companies are doing the same thing. If you go to the Medtronic website, you'll see something called Eureka, where they're asking for ideas from the outside. Innovative.com is something that came out of Lilly. They're doing the same thing. And then another example of something that we're doing called Data Design Diabetes is crowdsourcing to generate ideas. So let me just show you a moment on this. So we put out there that we would, in terms of crowdsourcing exercise, where you give an award for ideas. And in this case, we wanted something which would help us think about treating patients in the diabetes area different than they had been treated before. And ultimately came back with something which is actually somewhat similar, I think, what Don was talking about in terms of anticipatory medical devices, which is something called, from a company called Ginger.io, where this is something that can, it's an app for a phone that allows diabetes patients to understand whether or not there's a problem. If we don't see much activity with the diabetes patient, their activity drops down, then a signal is sent and to their healthcare provider or their spouse, just to warn them that something may be happening. Companies more and more are going to be going to this type of innovation paradigm to get ideas that allow them to jumpstart their product development and discovery efforts. This sounds great, but there's a lot of challenges. And I think some of you already know some of these challenges from a legal perspective. companies are infamous for having very complex legal processes for more open collaborations, but it's something that we are changing and we need to change to be able to bring in these ideas for us to ultimately transform into products. Similar types of challenges for intellectual property as well as for competition. But this is happening and it's starting to change. And then I mentioned the importance, and I won't belabor this because it's already been covered by Don, of the academic environment being one that is helping us in terms of these observations and turning these observations into things and more than just ideas, but concepts that can be reduced to practice. What companies are starting to do, and I'll stop after this slide, is to also change their innovation paradigm. What we've done at Sanofi is set up R&D hubs, and other companies are starting to do this as well. So you put these hubs in areas where you have highly creative, what we call ecosystems. In our case, we're in the Boston area. So we have MIT, Harvard, Boston University, the medical schools, other academic institutions. And we're purposely in those locations to try to do innovation in a different way, where we're not just doing things internally, but we're working with our local environment to try to develop these ideas. So ultimately, longer term, I think what you're going to see is that companies more and more are doing this type of innovation. And I think all of us, whether we're in industry or in academia, we're going to find that we have to collaborate in a different way for us to generate the ideas that ultimately become our innovative products of the future. Thank you very much. I know it's late, so if you have any questions, I'll be lingering a little bit, so I'd be happy to answer them.
Well, I want to thank all of our speakers. This has been extremely interesting and thought-provoking for us. I really appreciate Jim's comments from the industry perspective, this new model for collaboration and the new needs from the industry side. So thank you all for staying and for being here and participating, and our outstanding speakers. I also want to recognize Bob Neeram, who was my helper in putting this symposium together. So